Welcome back to the second part of our fungus lecture, where we're going to take a look at some of our major fungal phyla. In the first part of our lecture, we talked about general features that we saw in the fungal kingdom. This is a close-up of the branch of our tree of life that looks at fungi in particular, and we talked about the structure of fungi, including hyphae and mycelia. We talked about how cell walls consist of chitin, and we talked about some of the features of plant life cycles, including that they go through plasmogamy followed by karyogamy to complete the sexual portion of the life cycle. Now we're going to take a look at major phyla, and we're going to start with the Chytridiomycota, which are generally considered to be one of the more evolutionarily ancient phyla for reasons that we'll see in just a moment. We're then going to take a look at the Zygomycota, and I'm also going to briefly mention the Glomeromycota as well. These two life cycles both have similar reproductive structures, although they have different habitats. In particular, the Glomeromycota are going to be fungi associated with plant roots. They form these arbuscular structures that go into plant roots and receive carbohydrates in exchange for nutrients, such as phosphates and nitrates. Our third phylum is going to be the Ascomycota. And finally, we're going to take a look at the Basidiomycota, which might be the group of fungi that you are most familiar with. These would include the mushrooms. This is a version of the tree in your textbook as well, and notice the general similarities in terms of the phyla that we're talking about here and in the tree produced by Dr. Stevens. This first introductory table summarizes some of the unique features that we're going to see in each of our fungal phyla. So in our Chytridiomycota, we're going to see motile spores and, in fact, even motile gametes in some species, indicating or providing evidence for the ancestrality of this particular phylum. However, in many of our other phyla, what we're going to see is that the phylum name and, in fact, some of the key distinguishing features between these phyla have to do with a very specific reproductive structure. And this particular reproductive structure is the cell in which the zygote forms. So in our Zygomycota, we're going to see a zygosporangium. That's the cell where karyogamy occurs. In our Glomeromycota, they have this, as I mentioned, the same general reproductive structures as our Zygomycota, but we're going to see arbuscules in particular. In our Ascomycota, the sac fungi, we're going to see asci, and asci are where karyogamy occurs. And finally, in our Basidiomycota, our clump fungi, their cell where karyogamy occurs is the basidium. Early fossil evidence of fungi goes back at least 460 million years into the Ordovician. So this is similar to when we see the evolution of plants on land. For these fungi, in these fossils, notice that we can see various hyphae branching, as we do in all fungi. They also have sort of these very globular reproductive structures, and they seem to be aquatic, which would make sense because most of our major multicellular organisms originated in water and then made some sort of evolutionary transition to land. And this seems to be the case with the fungi too. As we look at the Chytridiomycota, we see a few features about this phylum that seem then to correspond to it being a more ancestral group of fungal species. In particular, most of our Chytridiomycetes are aquatic. And to go along with their aquatic habitat, they have what are known as zoospores. So all the spores that we've talked about so far, the spores that I drew in the fungus life cycle, weren't motile. They were small structures, but they couldn't actually move themselves. So zoospores are spores with flagella. They're actually capable of propelling themselves through the water. Why else would you need a flagellum on a spore unless the fungus is an aquatic group of organisms? So the Chytridiomycetes, for this reason, because they're aquatic, because their spores can swim, seem to be more ancestral. Here's a picture from your textbook of a modern chytrid fungus. Notice its at least superficial similarities to our fossil fungi. We see the hyphae clearly. We also see these large globular reproductive structures. One final note about the Chytridiomycetes is that they seem to, at least some species, cause harm to a number of amphibian species in fresh water. What happens is that particular species of chytrids can produce toxins in abundance, and these toxins can either kill amphibians outright, or they can cause developmental deformities, so loss of limbs, maybe an extra limb, a misplaced limb, and so on. These problems with chytridiomycetes seem to be getting worse as our climate warms, as temperatures are getting warmer around the globe. So warmer temperatures allow chytridiomycetes to reproduce more, and these higher reproductive levels contribute to higher toxin levels that then are harming amphibians. So current estimates are that something like a third of amphibian species are going to become threatened and endangered within the next century. And a lot of this threat and endangerment is going to have to do with global warming interacting in a very negative way with chytridiomycetes. Our second fungal phylum is the Zygomycota. So here is its life cycle, and we can see several features of the generalized life cycle, but let's take a look at a few points that distinguish this from a general fungus life cycle. First of all, let's start with spores. So we have a lot of one-end spores. The zygomycetes have an asexual portion of their life cycle. So the spores germinate, they produce hyphae, and we can see that this asexual reproductive structure is a sporangium. It looks at least superficially like the sporangia that we see in the plant kingdom. And then the sporangium ruptures, releasing a lot of spores. As these spores continue to grow, there can be different mating strains, as we saw. And when two hyphae of different mating strains come together, that's our process of plasmogamy. So plasmogamy in the Zygomycota is just going to involve a single cell. And this single cell is going to be eventually referred to as a zygosporangium. In the zygosporangium, what's going to happen is nuclei of these two different mating strains are going to come together to form zygotes, and in fact there can be a couple of zygotes, multiple zygotes formed inside the zygosporangium, but the zygosporangium is eventually going to develop sort of this rough exterior as we see right here. Then once we have our zygotes, they're going to undergo meiosis. So in this case we have three zygotes, they're undergoing meiosis. One of them will germinate to produce a sporangium that again releases spores, and our life cycle is now complete. If you want to think of examples of zygomycetes, you probably have seen these. A very common zygomycete would be bread mold. So here's a piece of bread. The little moldy cells that you see on it are in fact this fungi, is Rhizopus, and it has a little sporangia right here. You can see the hyphal structure is right there. But as frustrating or sometimes disgusting as we think Rhizopus and bread mold might be, a lot of Zygomycota are also very important decomposers, particularly decomposers, among other things, of dead animals or animal waste. So one particular species that, one particular genus that does that very well is the genus Pilobolus, that decomposes a variety of animal feces. This leads to the old fungal joke told by mycologists, particularly frustrated mycologists. People treat me like Pilobolus, they keep me in the dark, and they feed me horse manure, to put it politely. The third fungal phyla is the Ascomycota. And again, we see several features of the generalized fungal life cycle, but also several distinct features. Let's again begin with spores, or one-end spores, and they germinate to produce fungal hyphae of different mating strains. Like the Zygomycota, the Ascomycota are capable of asexual reproduction. So here is a hypha growing, but eventually it produces the structure that isn't a sporangium. Instead, it's a structure that doesn't enclose spores, but that bears spores at the tips of what we'll call branches, for lack of a better word. And so this structure is called a conidiophore. Phore because it's a bearing structure, conidia because that's what it holds. So the term for a single one-end spore-like cell in the Ascomycota is not spore, but conidium. Conidium is singular, conidia are plural. So here we see a conidiophore with lots of conidia. These conidia can be released, and then very much like spores, they can germinate to produce mating strains of their own. Our life cycle continues when mating strains of different types, a plus strain and a minus strain, come together and fuse in the process of plasmogamy. Unlike our zygomycetes, however, once plasmogamy occurs, these cells can then persist and divide in the N plus N, or dikaryotic stage, for a long time. In fact, so long that they produce a very large structure. Eventually, they're going to produce this ascocarp that we see over here. So an ascocarp is very much a visible structure. It can be big, think of, as we'll see in a moment, a morel mushroom, for example. It's not actually a mushroom, but it's a very big structure. Lots of N plus N cells. Ultimately, though, it's going to produce a cell called an ascus. And this ascus is where karyogamy is going to occur. So karyogamy is the fusion of the nuclei. We get two 1N nuclei that fuse to form a diploid zygote. Our diploid zygote is then going to undergo meiosis, and occasionally in species, what happens is these 4 cells that result, each of these then undergo mitosis to give us a total of 8 ascospores. So this last picture shows the whole ascocarp together. Here's the outer ascocarp. Here you can see individual asci. Inside each asci are these 8 ascospores, and the ascospores are being released to start our sexual life cycle over again. The ascomycota are known as the sac fungus because often what happens is that the ascus is sort of a, that the ascocarp is sort of a sac-like structure, a sac that's upright. However, some of the more common ascomycetes that you might be familiar with don't have sort of as clear a sac structure. So morels, what are known as morel mushrooms, even though they're not actually mushrooms, are ascomycota, as are truffles, if we want to think about some particularly rare and particularly tasty examples of this particular phylum. Our final fungal phylum is the Basidiomycota. And the Basidiomycota are probably best known to you, and think about mushrooms. Let's again start the life cycle with spores, 1N reproductive cells. The first and very interesting thing we notice about this life cycle compared to the others is that there isn't any kind of asexual reproductive loop over here. So it's not clear why basidiomycetes don't have this asexual reproductive part of their life cycle, but it seems that they don't. Spores are going to germinate, and they'll produce different mating strains. Eventually, if the mating strains come together, we have plasmogamy. Again, like our ascomycota, our basidiomycota can have a plasmogamous phase that persists for a long time and leads to the production of very large above-ground reproductive structures. In this case, what we're going to call a basidiocarp instead of an ascocarp, or what you might know as a mushroom. If we look at the undersurface of the cap of a mushroom over here, we'll see lots of gills. And if we zoom in on the gills, we see lots of these cells that are now called basidia. So this actually refers to club, and we'll see why this is these cells are club-like in just a moment. So inside each basidium, we start out with 2 1N nuclei in the N plus N phase. We go through karyogamy, or fusion of the 2 nuclei, to give us a zygote in each of our basidial cells. Our zygote then quickly undergoes meiosis, so we get 4 spores, otherwise known as 4 basidiospores per basidium. And so these spores then will make their way to the external tip of the cell. You can see them right here in this electron microscope picture, just before they're released. And so this basidium cell here with the 4 spores dangling off the tip of it looks sort of club-like, I guess, to a lot of early mycologists, and so they named this cell a basidium, and they named the structure that holds it the basidiocarp. So the spores are released, and we have started our life cycle again. Finally, please note that there are 4 basidiospores per basidium, not 8. I just caught that error and corrected it. Here are a couple of examples of different basidiomycetes. Mushrooms, of course, are one example, as we've been talking about. Another example are shelf fungi that often decompose dead, rotting trees. Sometimes they even get started before the tree is dead. So these are sort of laying on their side. What's happened is that the tree has fallen over, but initially these basidiomycetes, these shelf fungi, were more horizontal as opposed to being oriented vertically as we see in this picture. But what this picture shows is that there are lots of sort of pore-like structures on which our basidia can form on the underside of this shelf fungus, much like there are gills on the underside of a mushroom. One final feature you might notice about some of these fungi, such as mushrooms, is that they will form structures that are known as fairy rings when the basidiocarps emerge. So what's going on with this? What we have in the middle of this so-called fairy ring is this large mass of the mycelia of this particular individual. So all of these mushrooms over here have the same genotype, and they're emerging from a mycelial mass that has the same genotype as well. So this mycelial mass has been growing year after year after year, and each year it moves a little bit further out, and then it produces basidia at the ends of this mycelial mass. So the structure that we know as a fairy ring gets bigger and bigger and bigger each year. And ultimately, this mycelial mass can become enormous. So it's thought that some of the biggest multicellular organisms in the world are fungi that have been growing these mycelial masses not just for years, but perhaps even for millennia. Very neat when you think about it. Before we leave fungi altogether, I'd like to talk about a final symbiotic association that gives rise to a unique set of species known as lichens. So lichens are this association between a fungus and an algae. And so the algae can be a eukaryotic algal cell, or it can also be a cyanobacterial cell. And most of the algae that are part of this association are green algae, but recently it's been discovered that red algae can be part of the association as well. The fungi that form the association are often ascomycetes. So why would we get this association, this symbiotic relationship between a fungus and an algae? It seems that they're very different, you know. Algae tend to be aquatic, fungi tend to be terrestrial. How are these going to come together? So what the fungus gets from this are carbohydrates. So algae are photosynthesizing, in particular if the fungus can provide a slightly less dry, sort of ever so slightly a little bit more aquatic environment. For algae, the algae can persist, they can photosynthesize, they can provide carbohydrates to the fungus. In return, as I said, the algae gets a place that's a little less dry, and this is particularly important for green algae. If you remember our green algal lecture, we mentioned that some green algae can actually grow in some slightly more terrestrial environments, including even the sides of buildings. So this, for green algae that are a little bit more terrestrial, a lichen can provide a safer, less dry place for it. The lichen also can provide mineral nutrients, much as mycorrhizae can provide mineral nutrients for plants through their roots. If we take a look at the fungus and look at it in cross-section, we can see both the fungal hyphae sort of forming dense mats at the barrier. We can also see our algal cells mixed with the fungal hyphae. And so this mixing of the algal cells and the fungal hyphae give rise to the reproductive structures, at least the asexual reproductive structures of our lichen. So asexually, the reproductive structure is known as a soredium singular or soredia plural. Here it is in an electron microscope picture with some artificial coloring added. And this collection of hyphae wrapped around an algal cell will be released, and if it lands in an appropriate place, it will germinate and it will produce a new lichen. Lichens can also reproduce sexually, but in order to do that, each of the two species in the symbiotic relationship has to reproduce on its own. So here we see the ascocarp of a fungus and the ascocarp is going to produce ascospores inside the asci. Those would have to disperse and reproduce on their own. The individual algal cells would have to disperse and reproduce on their own too, and the two would have to get together to reestablish this symbiotic relationship. We can see lichens all around us in the world. They tend to be very inconspicuous, but ecologically they're very important. They're very good at colonizing spaces that few other organisms, fungal, algal, plant, can really colonize. And so they have different looking forms, in some cases based on the type of environment in which they're colonizing. So some lichens have a crustose form. In other words, they can just kind of really encrust themselves on rock, which is a very inhospitable place for multicellular organisms to live. A slightly more three-dimensional structure can be achieved by foliose lichens. So these are leaf-like lichens. They too can grow in some very inhospitable places, such as rock surfaces. Finally, we can have fruticose lichens. And fruticose lichens have the most three-dimensional structure. Fruit over here does refer to fruit. It sort of looks like more of a plant-like or fruiting sort of structure. It even looks maybe a little bit like Spanish moss. So here we see some fruticose lichens dangling down. We can also see fruticose lichens have their own three-dimensional structure and grow kind of like little plants or little shrubs. So I'll end by mentioning a particular type of lichen that I just love seeing here in Kentucky. Throughout the lectures, what I've tried to do are emphasize the organisms that we can see in each of these groups and some really interesting examples of these all over the world. But to end, I'd like to bring our examples back here to Kentucky, and remember that we can see some really neat examples of organisms right here in Kentucky. And in fact, Kentucky is an excellent place for seeing a diversity of species from all over North America. We get in Kentucky species that are sort of on the western edge of the range that tend to be more eastward. We also tend to get species that are sort of at the very easternmost end of Great Plains. They're prairie species. We tend to get species that are at the northern edge of a southern range. And finally, we can get some species that are at the southern end of a very northerly distribution. And that's what we have over here with our reindeer lichens. If you've had a chance to go hiking in Kentucky and get on top of ridges, such as mountain ridges in the Cumberlands, or maybe some of these sandstone plateaus or ridges in the Cumberland Plateau, sometimes you can come across a little area that looks like this. It sort of looks whitish, almost looks like it's snowing in the middle of the summer. And these are reindeer lichens. And the idea behind these little lichen patches is they actually seem to be a refugia left over from the last glaciation. So our most recent glaciation ended a little over 12,000 years ago, came down to a little bit north of where the Ohio River is. And so the Ohio River then forms from melted glacial water. But it bulldozes a lot of land to the north of it, which means that there wasn't a lot of habitat for organisms to be living there if that land is covered in ice sheets. So a lot of these boreal organisms, or organisms we now think of as living in northern parts of North America, had to migrate south to escape the glaciers. And so they could hang out on the Cumberland Plateau, the Appalachian Mountains that we have right here in Kentucky and in Tennessee. Then, as the glaciers retreated, these species that persisted in these little refugia began to move north again following the retreating glaciers. And reindeer lichen is an example of one of these species. So there are a number of species of reindeer lichen. They sort of have a circumpolar distribution now. So you can see them certainly in Canada and far northern places like Newfoundland. You can see them around the Arctic Circle, but we have them right here in Kentucky as a persisting species. So I think it's really neat that we can spend time in Kentucky and see examples of plants and examples of communities that are representative communities in very, very different parts of the world. And so that's the thought that I'd like I'd like to leave you with for all of these lectures as we think about the ecological significance of the species that we're looking at. Here in Kentucky, we're extraordinarily fortunate to see examples of species from all over North America, and in fact, all over the world. We're sitting on a biodiversity hotspot here in North America. So go out, enjoy it, take care of it, see neat areas like our little reindeer lichen patches, but other very neat habitats that we have all over our state. I hope you've enjoyed this lecture. I hope you've enjoyed all of our plant, fungal lectures, and I look forward to keep working with you as we explore the animal kingdom coming up next.